Cleaning validation is a methodology used to ensure that the cleaning process removes residues of the active pharmaceutical ingredients of the products manufactured in a piece of equipment. The cleaning aids utilized in the cleaning process and the microbial attributes. All residues are removed to predetermined levels to ensure that the quality of the next product manufactured is not compromised by waste from the previous product and the quality of future products using the equipment to prevent cross-contamination and as a GMP requirement.
Evaluation of cleaning validation. Equipment design. Examine the design of the equipment, especially in those large systems that may employ semi-automatic or fully automatic clean-in-place systems, since they represent significant concern. For example, stationary type piping without ball valve should be used. When such non-stationary ball valves are used, as is common in the bulk drug industry, the cleaning process is more difficult. A cleaning process written, procedure and documentation. Examine the detail and specificity of the procedure for the cleaning process being validated, and the amount of documentation required have been general SOPs, while others use a batch record or log sheet system. Analytical methods. Determine the sensitivity of the analytical method used to detect residuals or contamination. With advances in analytical technology, residues from the manufacturing and cleaning process can be detected at very low levels.
Sampling techniques. The following sampling techniques can be used for collecting the sample for the equipment. It consists of two techniques: range sampling and swab sampling. Range sampling allows sampling of the large surface area. In addition, an accessible area of equipment that can't be routinely disassembled can be evaluated. However, consideration should be given to the solubility of the contaminant. Swab sampling technique. For selection a suitable swab sampler, swab shall be compatible with product, allow extraction of the compound for analysis, swab shouldn't release fibers and shouldn't cause degradation of product. And for preparation, select a suitable swab, define area to be tested, and dampen swab with diluent. The sampling consists of 4 steps. In step 1, swab with overlapping pattern. In step 2, swab and passing swab in the perpendicular direction. And in step 3 and 4, repeat procedure with second swab at 45 angle. Before transfer for analysis, snap swab head at notch long edge of a swab handle. Then allow the head to fall into the vial. Finally, transfer for TOC or HPLC for analysis.
I will talk about detergent. Detergents released by quality control and meet food standards or regulation. Composition known. Easily removed with rinsing, demonstrated with acceptable limits defined. If persistent residue, example cationic detergent, avoided, consider also detergent breakdown. I will talk about basic principle of the GMP. Which are the critical area for sampling? What would be considered an appropriate approach for cleaning validation for a piece of equipment? Then, I will talk about cleaning requires. Product manufactured previously, product manufactured afterward, characteristics of manufacturing process, characteristic of the equipment used, and characteristic of the cleaning process. I will talk now about recovery. Recovery, if it's more than 80%, it's considered good. More than 50% is considered reasonable. Recovery less than 50% is considered questionable.
I'll speak about conclusion. It is practically impossible to prove that production equipment is clean at the level of 100%. However, it is possible to prove that the traces of active product remaining spread through the equipment part are within an acceptable limit, and that we are capable of detecting and quantifying these trace levels. Cleaning validation provides a mean of providing that the contamination levels have been reduced below contamination acceptance limits. It is concluded that to control the carryover of leftover residue from previous batch to the next batch, an effective validated cleaning mechanism shall be in place. This shall contain defined cleaning procedures. Number 2, cleaning validation policy. Number 3, a validation protocol. Number 4, validated chemical and microbiological methods.